Hello friends, welcome back to a new video that is MCQs on biophysical techniques and biological methods. And this is the part 2 of this series of videos on biophysical techniques and biological methods. Okay, so the first question of this video, that is the polymerase chain reaction or PCR, is a technique that option A was used to demonstrate DNA as the genetic material. Option B is used to determine the content of minerals in a soil sample. Option C uses short DNA primers and a thermostable DNA polymerase to replicate specific DNA sequence in vitro. Or option D, it measures the ribosome transfer rate during translation. So which one is correct about the PCR or polymerase chain reaction? So the right answer is option C. That is, this PCR technique uses short DNA primers and a thermostable DNA polymerase to replicate specific DNA sequence in vitro. Okay, so this is known as polymerase chain reaction or PCR. So let's move on to question number 22. That is, a major problem with the computational assembly of DNA sequences of complex eukaryotic genomes is the presence of option A, multiple chromosomes, option B, mitochondrial DNA, option C, introns with the genome, or option D, repetitive sequence. And the right answer is option D, that is repetitive sequence. So a major problem with the computational assembly of DNA sequences of complex eukaryotic genomes is the presence of repetitive sequences. Okay, so let's move to question number 23. It is, what is the purpose of performing a homology search with a DNA sequence? And options are option A, to determine if any genes with similar sequences are present in the DNA databases. Option B, to determine if the sequence is already in the DNA databases. Or option C, to search for consensus exon intron boundaries. Or option D, to determine the codon bias for a specific gene. So the right answer is option A, that is to determine if any genes with similar sequences are present in the DNA databases. So what is the purpose of performing a homology search with a DNA sequence? That is to determine if any genes with similar sequences are present in the DNA databases or not. Okay, so this is the purpose of homology search with DNA sequence. So let's move on to question number 24. That is, in UV visible spectrophotometry, wavelength shift in bathochromic and hyperchromic when option A, the absorption maximum is shifted to a higher wavelength and its intensity does not change, or option B, the absorption maxima is shifted to a higher wavelength and its intensity decreases, or option C, that is the absorption maxima is shifted to a lower wavelength and its intensity increases, or option D, the absorption maxima is shifted to a higher wavelength and its intensity increases. So the right answer is option D, that is the absorption maximum is shifted to a higher wavelength and its intensity increases. So in UV visible spectrophotometry, a wavelength shift is bathochromic and hyperchromic when the absorption maximum is shifted to a higher wavelength and its intensity increases. Okay, so that is the option D is the correct one. So let's move on to question number 25 about the chromosome walking is best described as option A, aligning DNA sequences by computer to generate contigs, or option B, that is generating a map along a chromosome in a step-by-step manner. Option C, identifying clones whose inserts overlap to generate a library of clones that cover a given segment of DNA. Or option D, sequencing a genome at a time to ensure that no gaps are present at the end of the project. So which one is right about the chromosome walking? So the right answer is option C. It is identifying clones whose inserts overlap to generate a library of clones that cover a given segment of DNA. So this is known as chromosome walking. So let's move on to question number 26. Raise. So what could be the explanation for the fact that the yeast hexokinase does not work at 55 degrees Celsius but the recombinant hexokinase does? So what is the reason behind it? So options are option A, the recombinant hexokinase is more stable because it is being expressed from a plasmid. Option B, the recombinant hexokinase is from a bacterial origin and that makes it more functionally reliable at higher temperature. Or option C, the recombinant hexokinase probably has more disulfide bridges in its structure. Or option D, that is the yeast enzyme is less stable because yeast does not perform glycolysis to the extent bacteria do. So what is the right answer about the fact that the yeast hexokinase does not work at 55 degrees Celsius but the recombinant hexokinase does? So the right answer is option C, that is the recombinant hexokinase probably has more disulfide bridges in its structure, that's why it is more resistant to high temperature like 55 degrees Celsius. Okay, so this is question number 26. Let's move on to question number 27, and that is what electronic transition are the most important in UV spectroscopy of organic molecules because they result in the absorption of longer wavelength radiation outside the UV region. And the option A is sigma to pi and n to pi transition, or option B sigma to sigma star and n to sigma star transition, or option C pi to pi star and n to n star transition, or option D pi to pi star and n to pi star transition. So the right answer is option D, that is pi to pi star and n to pi star transition is more favorable. So what electronic transition are the most important in UV spectroscopy of organic molecules because they result in the absorption of longer wavelength radiation outside the UV region? That is option D, that is pi to pi star and n to pi star. Okay, so let's move on to question number 28. That is a microorganism when viewed under a compound microscope with objective of 40x and eyepiece of 10x magnification measured 400 micron in length. So the same microorganism when viewed under a dissecting microscope with 10x lens would measure option A 100 micron, option B 10 micron, option C 40 micron, or option D 400 micron. So when we observe in compound microscope, the objective is 40 and this eyepiece is 10. So total magnification is equals to 400 magnification. So by this 400 magnification, the microorganism's length is 400 micrometers. So when the magnification is 10x, so that time we will see that microorganism. So total magnification is 10. So what will be the length? That will be 10 micron. So the right answer will be option B, that is 10 micron. Okay, understand? So next question देखेंगे. Next is 29. That is the DNA mixture containing unlabeled and N15 labeled DNA from phage T4 was denatured and allowed to reannul. So how many bands one would observe upon cesium chloride density gradient centrifugation of the above mixture? Option A is 0 band, option B 1 band, option C 2 band, option D 3 bands. So the right answer is option D, that is 3 band we will get. So the DNA mixture containing unlabeled and N15 labeled DNA from phage T4 was denatured and allowed to reannul. So how many bands one would observe upon cesium chloride density gradient centrifugation on the above mixture? That is 3 bands. Okay, so let's move on to question number 30. It is, why did the researcher freeze the leaves before preparing the homogenate? And the options are option 1, the proteins are best preserved from degradation at low temperatures. Option 2, that is the water in the vacuoles needs to be frozen before homogenization. Option 3, that is plant tissue are easier to homogenate when they are frozen. Or option 4, that is ice crystals help sharing the cell walls. So which combination of these options is the correct one? So option A is 1 and 2 is correct, option B 2 only is correct, option C 3 and 4 is correct, or option D 2 and 3 is the correct one. So the right answer is the option C, that is 3 and 4 statement is the correct. So the plant tissue are easier to homogenate when they are frozen, and ice crystals help sharing the cell wall. So that's why the researcher freeze the leaves before preparing the homogenate. Okay. So next question देखेंगे. Next is question number 31. So a cell suspension was subjected to membrane disruption followed by differential centrifugation to fractionate the cellular components. So match the centrifugal conditions in column 1 to the appropriate subcellular components in column 2. So you have to match these two segments and we have to answer according to this option. So which option is the right? So let's see, the option A is the correct one. So here the P3, P is equals to matched with 3. That means P is matched with 3, that is at 1000 G for 10 minutes we will separate nucleus or nuclei. Okay, and by Q equals to 4, that means by 20,000 G RPM or gravitational force for 30 minutes centrifugation we will get 4, that means lysosomes and peroxisomes. Okay, and then R is 1, so by 80,000 G for 1 hour we will get microsomes and small vehicles, and by 1,50,000 G for 3 hours we will get the ribosomes. Okay, so by different gravitational force or different RPM with different time we will get different organelles. We can separate different organelles, okay, or precipitate down them. So let's move on to next question, that is question number 32. So DNA sequencing by the Sanger method takes advantage of which property of DNA synthesis to generate a sequencing ladder? And the options are option A, addition of nucleotides requires a free OH group on the 3' of the DNA strand. Option B, DNA has a free 5' phosphoryl group. Or option C, DNA polymerase has proofreading capabilities. Or option D, U is a base only found in RNA. So which is the property of DNA synthesis to generate a sequencing ladder? So by DNA sequencing by Sanger method takes advantage of which property of DNA synthesis? So the right answer is option A, that is addition of nucleotide requires a free OH group on the 3' of the DNA strand, and that advantage we use in DNA sequencing technique. Okay, so let's move on to question number 33. It is, what would happen if the concentration of dideoxynucleotides was too high in a chain termination sequencing reaction? And the options are option A, the reactions would yield very long molecules and there would be little sequence data close to the primer. Or option B, the reactions would yield very short molecules. Or option C, the reactions would not proceed as the high concentrations of the dideoxynucleotides would inhibit the DNA polymerase. Or option D, the fluorescence of the sequencing product would be too high and difficult to read. So the right one is option B, that is the reaction would yield very short molecules. So what would happen if the concentration of dideoxynucleotides was too high in a chain termination sequencing reaction? So reaction would yield very short molecules. Okay, so let's move on to question number 34. That is, 4 identical PCR reactions were carried out in tubes named 1, 2, 3 and 4. Beside the usual mix of DNTPs, each of the tubes respectively contained gamma P32, that means in the P32 gamma positions the isotope is present, radio labeled isotope is present of gamma P32 D ATP, then that is present in the tube number 1. In tube number 2, beta P32 T ATPs are present, and tube number 3, alpha P32 D ATPs are present, and in tube number 4, alpha P32 RNTPs are present. So which one of the tube will have radio labeled PCR product? So the options are option A tube 1, option B tube 2, option C tube 3 and option D tube 4. So we will get radio labeled PCR product when the radio labeled ATP will enter into the DNA strand. So radio labeled ATP will enter with DNA strand with the alpha position of the DATPs. So the right because in the ATP the 3 phosphate groups are attached one by one. Okay, so the first is the alpha, next is the beta, third is the gamma. So in the DNA strand, the alpha phosphate will enter. So we will get the radio level isotope of alpha P32 DATPs. Okay, so which is present in the tube 3. So the right answer will be C, that is tube 3. Okay, so next question देखेंगे. Next is question number 35. So in what cycle of PCR is a double stranded fragment of the correct size first generated? In what cycle of PCR is a double stranded fragment of the correct size first generated? And the options are option A second cycle, option B last cycle, option C third cycle, or option D 10th cycle. So in which cycle we will get the perfect double stranded DNA which we want to amplify? So the right answer is option C, that is in third cycle we will get the exact particular size amplified DNA fragment. Okay, so next question देखेंगे. Next is question number 36. So primer, sorry, so a primer with the sequence, this is the sequence of primer, is to be used in a PCR reaction. So using the formula that is Tm equals to 2 degree into A plus T, number of adenine plus number of thymine plus 4 degree into number of guanine plus number of cytosine. So what is the Tm of this oligonucleotide? So we have to calculate the number of A and number of T is present. So number of A and T is, number of A is 1, 2, 3, 4, 5 and 6. So A is 6. So T is 1, 2, 3, 4, 5 and 6. Here is also 6. So 6 plus 6, 12 plus 2 into 2 is 24. Okay, 24 plus here 4 degree into G plus C. G is G, total G is 1, 2, 3. Here is G is 3 and C is 1, 2, 3 and 4. That is total is 7. So 7 into 4, 28. So total will be 52. So the Tm would be 52 degrees Celsius. So the right answer is option B. Option B is the right answer. Okay, so let's move on to question number 37. That is, the region of DNA shown below is to be amplified by PCR. The appropriate pair of primers to use is option A, that is B and C, option B, that is A and D, option C, that is A and C, or option D, that is B and D. So the right answer is option A, that is B and C is the right answer. So that is B and C is the right appropriate pair of primers to use for amplifying the DNA by PCR technique. Okay, so let's move on to question number 38. That is, in selecting a DNA sequence to be used as a template for construction of PCR primers, many investigators look for sequences rich in codons for tryptophan or methionine because option A, tryptophan and methionine have only one codon each, that is, there is no oval. Option B, tryptophan and methionine have three codon each, or option C, codons for tryptophan and methionine abundantly present in mRNA, or option D, none of the above. So which one is the correct answer? The correct one is the option A, that is tryptophan and methionine have only one codon each, so that's why there is no oval is present. So in searching a DNA sequence to be used as a template for construction of PCR primers, to generate a PCR primer, many investigators look for sequence rich in codon for tryptophan and methionine because tryptophan and methionine have only one codon each. Okay, so let's move on to question number 39. That is, a nuclear magnetic resonance, that is NMR spectroscopy, the absorption spectra result by the absorption of one of the following electromagnetic radiation by the spinning nucleus is option A, ultraviolet waves, option B, infrared waves, option C, radio waves, or option D, microwaves. So the right one is option C, that is radio waves. So in nuclear magnetic resonance spectroscopy, the absorption spectra result by the absorption of one of the following electromagnetic radiation by the spinning nucleus is radio wave. Okay, so the last question of this video, that is, after isolating and purifying to the homogeneity of small enzyme, that is 110 amino acid long, from a culture of bacteria, you are confused as to whether you grew wild type bacteria or a mutant strain that produced the enzyme with a valine residue at position 66 instead of glycine found in the wild type strain. So for quick determination of the nature of the protein, you will use option A, mass spectroscopy, or option B, ion exchange chromatography, or option C, SDS page, or option D, HPLC. So which procedure you will follow to identify the nature of protein? So the right answer is option A, that is mass spectroscopy. So we will use mass spectroscopy for quick determination of the nature of the protein you will use. Okay, so these are all 20 questions of this part 2 video of on biophysical techniques and biological methods. Okay, so thank you for watching this video.